Hello everyone, I am Dr. Trupti. Welcome to my YouTube channel Enjoy Biochemistry. In today's video, we are going to learn about folic acid. At the end of this session, students shall be able to describe folic acid with respect to its sources, RDA, metabolism, biochemical functions and deficiency manifestations. Folic acid is also called as vitamin B9. It is the water-soluble vitamin and it is the hematopoietic vitamin because it is required for effective erythropoiesis. The erythroblasts require folate along with vitamin B12 for proliferation during their differentiation. And this folic acid has a role in purine synthesis as well as thymidylate synthesis, which is in turn required for the DNA synthesis. So if there is folic acid deficiency, it leads to ineffective erythropoiesis. The word folic acid is derived from the Latin word folia, which means leaf, and it has been isolated from the green leafy vegetables. The chemical name is pteroyl glutamic acid, and this pteroyl glutamic acid, it is made up of pteridine plus para-aminobenzoic acid and glutamic acid. So this is the pteroyl glutamic acid. It is the chemical name of folic acid. If we take a look at structure of folic acid, this is the pteridine, which is linked to para-aminobenzoic acid. Together this pteridine and PABA, it is called as pteroic acid. And when this pteroic acid is linked to one glutamic acid, it becomes pteroyl glutamic acid or folic acid. So it is the monoglutamate form which is present in the fortified food and dietary supplements. Humans can neither synthesize PABA nor attach glutamic acid residue to pteroic acid, and that's why human cannot synthesize folic acid. In nature, polyglutamate form is present, and in the polyglutamate form, these glutamic acid residues are about 2 to 7, and these 2 to 7 glutamic acid residues are linked to this pteroic acid in the polyglutamate form. And in the dietary sources, that is the natural diet, polyglutamate form is present. Now what is the active form of this folic acid? So tetrahydrofolate, which is the reduced form, it is the active form of or coenzyme form of folic acid, and the major form which is present in circulation is N5-methyl tetrahydrofolate. Now what is the difference between folic acid and folate? Are these two terms same or different? Sometimes these terms are used interchangeably, but they are different from the dietary perspective. The term folate should be used for bioactive forms of folic acid, for example dihydrofolate and tetrahydrofolate. What are the dietary sources of folic acid? Yeast, green leafy vegetables, animal liver, these are the rich sources of folic acid. So it is derived from both plant sources as well as animal sources. And the important plant sources are green leafy vegetables like spinach, broccoli, avocado, various citrus fruits. And the animal sources are eggs, animal liver, beef liver. And in these dietary sources, the folic acid is present in the polyglutamate form. And this folic acid is also available in the fortified food like flour, ready to eat breakfast cereals and bread, and here it is present in the monoglutamate form. And that's why it has a higher bioavailability than naturally occurring folate. Because this folic acid which is present in the folate supplements and in the fortified food, it is the poor substrate for polyglutamate synthase. What is the action of this enzyme? This is responsible for conversion of monoglutamate form into polyglutamate form, and this monoglutamate form can be efficiently taken up by the intestine and released to the blood, and that's why this monoglutamate form present in the fortified food and folate supplements, it has a higher bioavailability. What is the recommended dietary allowance for the folic acid? As we know that humans cannot synthesize folic acid, it has to be provided either through the diet or through folic acid supplements. Adult male and female, they require about 200 micrograms of folic acid per day. In pregnancy, the need is increased. It is about 400 microgram per day. Why there is increase in the need of folic acid in pregnancy? Because of increase in number of rapidly proliferating cells which are present in the blood, and this need is doubled in the third trimester. So if In pregnancy, folic acid is not supplied. It is linked to the condition like neural tube defects. In lactation, 300 micrograms per day, and if there is folic acid deficiency, then the dose is 1 milligram per day orally. Now, can this folic acid be stored in the body? Yes, it can be stored in the liver and kidney. About 5000 microgram is stored, which is enough for 2 to 3 months. And as it is only enough for 2 to 3 months, the folic acid deficiency manifests very early. Now let's see the absorption and metabolism of folic acid. When large doses of various folates are given, they can appear unchanged without any reduction or methylation in circulation because they are directly absorbed by the mucosal cells and sent into the circulation and readily taken up by the tissues like liver. And some of the folic acid is reduced to dihydrofolate by dihydrofolate reductase, and this reaction requires NADPH. Again, this dihydrofolate is converted to tetrahydrofolate by dihydrofolate reductase. In dietary sources, folate is present in the polyglutamate form having 2 to 7 glutamic acid residues attached to the steroic acid, and this folate glutamate cannot be readily absorbed in the intestinal mucosal cell. It has to be converted into folate monoglutamate. So the glutamate residues are cleaved off by polyglutamate hydrolase or folate hydrolase enzyme, and then this folate monoglutamate, it can be absorbed in the intestinal mucosal cells and converted to the reduced form, that is tetrahydrofolate. Further, the methylation of this tetrahydrofolate occur in the intestinal cells and it is converted to N5-methyl tetrahydrofolate, which can be absorbed in the monoglutamate form directly in the intestinal mucosal cells, and later it enters the circulation and then it reaches the various rapidly proliferating cells as well as tissues like liver and kidney for storage. And the folic acid which is taken up by the liver cells is converted to dihydrofolate, tetrahydrofolate and methyl tetrahydrofolate. This methyl tetrahydrofolate is a monoglutamate form which is converted to polyglutamate tetrahydrofolate because it is stored in the tissues like liver in the form of polyglutamate tetrahydrofolate, and this reaction occurs with the help of polyglutamate synthase enzyme. And whenever the tetrahydrofolate is required, then this polyglutamate tetrahydrofolate by action of enzyme folate deconjugates the glutamic acid residues are cleaved off, and again the methyl tetrahydrofolate can be released in the circulation and can reach to the various tissues and cells where the folic acid is required. 80% of circulatory folic acid is in the methyl tetrahydrofolate monoglutamate form and 20% is in the other forms like tetrahydrofolate and formyl tetrahydrofolate. Coming to the biochemical function of folic acid is involved in the 1-carbon metabolism. It is the carrier of 1-carbon groups. Now what are those 1-carbon groups? The various 1-carbon groups are methyl group, methylene group, methinyl group, formyl, form imino and hydroxymethyl group. Now all these 1-carbon groups, they are carried by various tetrahydrofolate. For example, N5-methyl tetrahydrofolate carries methyl group, N5-N10 methylene tetrahydrofolate carries methylene group, N5-N10 methinyl tetrahydrofolate carries methinyl group, N10 formyl tetrahydrofolate carries formyl group, N5 form imino tetrahydrofolate carries form imino group, and N10 hydroxymethyl tetrahydrofolate is a carrier of hydroxymethyl group. And now all these 1-carbon groups, they are important for DNA or RNA synthesis. C2 and C8 of purines require this 1-carbon group. Thymidylate synthesis, which is ultimately required for the DNA synthesis. Here the 1-carbon is carried with the help of these tetrahydrofolates. It is also required for the amino acid metabolism, that is interconversion of serine and glycine, histidine catabolism, methionine synthesis from the homocysteine, then synthesis of choline ethanolamine. So in the amino acid metabolism, this 1-carbon group is very important. In addition to that, protein biosynthesis requires the initiation of protein biosynthesis starts with N-formyl methionine, that is the initiator of protein biosynthesis, and it is formed with the help of this 1-carbon group. So these are the various roles of 1-carbon group in DNA RNA synthesis, amino acid metabolism, and protein biosynthesis. And most of the 1-carbon transfers are promoted by tetrahydrofolate and S-adenosyl methionine. Now let's see the various sources and utilization of tetrahydrofolates, various forms of tetrahydrofolate. So as we have seen, there are various forms of tetrahydrofolate. For example, N10-formyl tetrahydrofolate, N5-N10 methinyl tetrahydrofolate, N5-N10 methylene tetrahydrofolate, and N5-methyl tetrahydrofolate. So let's see how they are formed and how they are utilized further. So this N10-formyl tetrahydrofolate, it is derived from the glycine and tryptophan metabolism, where formate is formed, and it results in this N10-formyl tetrahydrofolate, and further it is utilized for the C2 of purine synthesis, and it is also involved in the synthesis of formyl methionine. This N5-N10 methinyl tetrahydrofolate is derived from the histidine catabolism. On catabolism of histidine, FIGLU is formed, and then THF tetrahydrofolate is incorporated to form N5 form imino tetrahydrofolate, and ultimately we get N5-N10 methinyl tetrahydrofolate, which is utilized for the purine synthesis. C8 of purine is derived from this N5-N10 methinyl tetrahydrofolate. Then interconversion of serine and glycine, it involves N5-N10 methylene tetrahydrofolate, and this N5-N10 methylene tetrahydrofolate, it is required for the thymidylate synthesis. That is, thymidylate is formed from the deoxyuridine monophosphate. Then the choline and betaine metabolism is involved in the formation of N5-methyl tetrahydrofolate. And this N5-methyl tetrahydrofolate, it is the predominant form of tetrahydrofolate in the circulation. And further, this N5-methyl tetrahydrofolate is converted to tetrahydrofolate, and which involves the formation of methionine from homocysteine, which requires vitamin B12. In this process, the methyl group is released, and which is involved in the various transmethylation reaction. So the tetrahydrofolate, it is present in the body in two pools. One is active pool and other is the reduced pool. So in the reduced pool, N5-methyl tetrahydrofolate is the reduced pool of tetrahydrofolate, and that is the major form of folate which is absorbed from the small intestine, that is most importantly jejunum, and it is the major form which is circulating in the blood for transport to the various tissues. And the active pool involves the other tetrahydrofolate other than this N5-methyl tetrahydrofolate, and the active pool forms of tetrahydrofolate, they can be converted to 5-methyl tetrahydrofolate, but 5-methyl tetrahydrofolate cannot be used to synthesize active pool of other tetrahydrofolate derivatives. So it is like a dead end, that is N5-methyl tetrahydrofolate cannot be converted to other forms of tetrahydrofolate. Now let's see what is the role of vitamin B12, that is cobalamin, in the folic acid metabolism, and what is folate trap. So we know that this methionine, it is converted to S-adenosyl methionine, which is the active methyl donor, by action of enzyme methionine adenosyl transferase, and this reaction requires ATP. Further, this S-adenosyl methionine is converted to S-adenosyl homocysteine. So here methyl group is donated to the acceptor, and methylated product is formed. For example, the formation or methylation of norepinephrine to epinephrine, and this reaction occurs with the help of enzyme methyl transferase. This S-adenosyl methionine, it is the active donor of methyl group. Further, this S-adenosyl homocysteine, it is converted to homocysteine by enzyme adenosyl homocysteinase. Now, the regeneration of methionine from this homocysteine, it requires vitamin B12 as well as folic acid, and the enzyme methionine synthase or homocysteine methyl transferase, because the homocysteine requires methyl group so that methionine can be formed from this homocysteine. Now how this methyl group is um, get incorporated in the homocysteine so that methionine is formed, let's see. So this reaction requires cobalamin as well as N5-methyl tetrahydrofolate. Here the methyl group from this N5-methyl tetrahydrofolate is donated to this vitamin B12, that is cobalamin, and there is formation of methyl cobalamin and the tetrahydrofolate is released. Now this methyl cobalamin, it gives its methyl group to the homocysteine to form methionine, and then cobalamin is regenerated. So the formation of methionine from homocysteine requires vitamin B12 as well as N5-methyl tetrahydrofolate. Now what is folate trap? So imagine if there is deficiency of vitamin B12 or if there is defect in the enzyme methionine synthase, then this N5-methyl tetrahydrofolate cannot donate its methyl group to cobalamin to form methyl cobalamin, and further this methyl group cannot be donated to homocysteine to form methionine. And in this deficiency of vitamin B12 and methionine synthase, folate will be trapped in the form of N5-methyl tetrahydrofolate, which cannot be converted into any other forms of tetrahydrofolate. And that's how in vitamin B12 deficiency it is manifested as a folic acid deficiency because of this folate trap. So this is the important role of vitamin B12 in the folic acid metabolism. What are the various causes of folate deficiency? Decreased dietary intake, decreased absorption of folate, increased demand of the folate, and decreased absorption because of various drugs. So these are the various causes of folate deficiency. And malnutrition is the cause of dietary deficiency, that is reduced intake or poor nutrition in case of elderly patient also. And alcoholism is also important reason for poor nutrition. It also decreases the absorption of folate, and this dietary deficiency is the most common cause of folate deficiency. When there is decreased absorption of folate occurs, it can occur in the various intestinal disorders like sprue celiac disease, gluten-induced enteropathy, or whenever there is resection of jejunum, because the absorption of folate occurs mainly in the jejunum. And which are those conditions when the folic acid demand is increased and it results in the folate deficiency? In the pregnancy, because of rapidly proliferating cells, the demand is increased. In the lactation also it is increased. In various malignancies, because of increased cell turnover, in chronic hemolytic anemia, the demand of folic acid is increased, and that's why it results in the folate deficiency. Various drugs like phenytoin, hydantoin, anticonvulsant drugs and also the sulfasalazine, they interfere the absorption of folic acid. In addition to that, certain folic acid analogues like methotrexate also interferes with this formation of tetrahydrofolate, and it is also the important cause of folate deficiency. What are the important deficiency manifestations of folic acid? It results in the deranged DNA synthesis, because folic acid is required for the DNA synthesis. It is involved in the purine and thymidylate synthesis, and that's why if there is lack of folic acid, it will result in the defective DNA synthesis. Then neural tube defects are also associated with the folic acid deficiency. If in the pregnancy there is increased demand of the folic acid, and if it is not provided, it can result in the various neural tube defects. Hyperhomocystinemia is also a result of deficiency of folic acid, because the homocysteine to methionine generation requires folic acid, and if the folic acid is deficient, then homocysteine cannot be converted to methionine, and that's why it results in the increased homocysteine level in the blood. Now this deranged DNA synthesis can be manifested as a macrocytic anemia with the megaloblastic changes. The various neural tube defects can be spina bifida, anencephaly, and encephalocele. So these are the various neural tube defects associated with the folic acid deficiency. Hyperhomocystinemia is associated with the atherosclerosis, which ultimately leads to the coronary artery disease. What is the biochemical basis of macrocytic anemia in folate deficiency? We know that methylene tetrahydrofolate is required for the thymidylate synthesis. So if there is folate deficiency, there will be decreased thymidylate. And there is cell cycle arrest in the S phase of rapidly proliferating cells, which results in the defect in the DNA synthesis. So here the nucleus stop maturation, but cytoplasm maturation and HB synthesis continues. So there is asynchronous maturation between cytoplasm and nucleus, and because of this there is a formation of abnormally large erythrocytes which are called as macrocytes. And there are megaloblastic changes are also seen here due to folic acid deficiency. These megaloblasts are the large nucleated red blood cells precursors with non-condensed chromatin due to impaired DNA synthesis. So macrocytic anemia due to folic acid deficiency is a megaloblastic anemia. The another theory is like that if there is decreased thymidylate synthesis, then this lack of thymidylate leads to incorporation of uracil in the replicating DNA strands, and the repair enzymes find this defect of incorporation of uracil, try to correct it unsuccessfully, and the newly formed DNA strands are destroyed by apoptosis, and thus it also results in the defective DNA synthesis. Now let's see the clinical features and laboratory findings in the megaloblastic anemia due to folic acid deficiency. Peripheral blood smear shows macrocytosis, that is the RBC size is increased and hypersegmented neutrophils are seen. Bone marrow examination shows megaloblasts, which are the immature enlarged RBC precursors. Anemia is present because of ineffective erythropoiesis. Splenomegaly is seen because abnormal RBCs are destroyed in the spleen, and it leads to increased bilirubin level because of hemolysis, and it shows pale yellow appearance. Various clinical features which can be seen in megaloblastic anemia due to folic acid deficiency. In such cases, history is very important because the patient can present with the excessive alcohol intake, and we know that alcohol intake is associated with the poor nutrition, and absorption of folic acid is also retarded. Use of various drugs is also important because it can decrease the absorption of folic acid. Then patient can present with various oral lesions like sore, red, swollen tongue, angular stomatitis can be present. Various gastrointestinal symptoms can be the manifestation of megaloblastic anemia like nausea, vomiting, abdominal pain, diarrhea, and hyperpigmentation may also be found on the dorsal surface of finger and toes. Megaloblastic anemia can occur due to deficiency of both folic acid as well as vitamin B12. So it is very important to know whether the megaloblastic anemia is due to folic acid deficiency or vitamin B12 deficiency or due to deficiency of both. So in case of folic acid deficiency, folic acid level is reduced. So, but the vitamin B12 level will be normal in folic acid deficiency, while in case of vitamin B12 deficiency, it will be low. Now, the methylmalonic acid level in the blood will be normal in folic acid deficiency, but it will be elevated in vitamin B12 deficiency, and homocysteine level will be elevated in both the condition. So that's how by estimating vitamin B12 level and the methylmalonic acid level, we can find out whether the megaloblastic anemia is due to folic acid deficiency or because of deficiency of vitamin B12 or due to deficiency of both. Other than megaloblastic anemia, neural tube defects is also important deficiency manifestation associated with the folic acid deficiency. We know that in pregnancy folic acid demand is increased, and if there is lack of folic acid during pregnancy, it results in the neural tube defects because due to folic acid deficiency, neural stem cell proliferation is hampered, and it results in the various conditions like spina bifida. In the spina bifida, spine and spinal cord don't form properly. Anencephaly, here the baby is born with an underdeveloped brain and incomplete skull. Encephalocele is a condition where a sac-like protrusion or projection of the brain and the membrane that cover it through an opening in the skull. So these are the three important neural tube defects associated with the folic acid deficiency. The third important condition associated with the folic acid deficiency is hyperhomocystinemia. Because folic acid is required for regeneration of methionine from homocysteine, and if it is deficient then it results in the increased homocysteine level, which is associated with the atherosclerosis and in turn leads to coronary artery disease. We know that lysyl oxidase enzyme converts lysine to hydroxylysine, which is important for collagen cross-linking and maturation. So now how this increased level of homocysteine is associated with the atherosclerosis? This homocysteine decreases the activity of this lysyl oxidase. It interacts with the lysyl residues of collagen and interfere with the collagen cross-linking, and thus it leads to abnormalities of collagen in the vessel wall. The second mechanism is that the homocysteine forms homocysteine thiolactone, which is a free radical which thiolates LDL particle. And these particles tend to aggregate and endocytose by the macrophages, and that's how it leads to increased tendency of atherogenesis. So in turn we can say that increased homocysteine level will increase the chances of atherogenesis or atherosclerosis, and which in turn can develop coronary artery disease. Let's revise the laboratory findings in folic acid deficiency. So we know that peripheral smear shows macrocytosis and hypersegmented neutrophils. Blood folic acid level will be decreased. The normal serum folic acid level is 3 to 16 nanogram per ml. It is reduced. Erythrocytic folic acid level is 130 to 630 nanogram per ml. RBC folate levels are the useful index of body stores of folate, and that is used to assess the duration of deficiency. Here, due to ineffective erythropoiesis and the hemolysis, there will be increased indirect bilirubin. LDH level is increased due to the hemolysis. Homocysteine level will be increased due to deficiency of folic acid. Histidine load test can also be done to detect the folic acid deficiency, because we know that catabolism of histidine leads to formation of form iminoglutamic acid, which is also called as FIGLU, and which is later converted to methinyl tetrahydrofolate. So if there is a folate deficiency, then this form iminoglutamic acid FIGLU, it cannot be converted into methinyl tetrahydrofolate and it is excreted in the urine. So earlier this test was being done, that is the histidine load test, but nowadays it is rarely done. What is the management of folic acid deficiency? It is oral folic acid is given to the person having folic acid deficiency, that is about 1 to 5 milligram daily. If there is intolerance to the oral folic acid, then IV, subcutaneous or intramuscular formulations can be given. If there is concomitant B12 deficiency along with the folic acid deficiency, then both should be given, both folic acid and vitamin B12. Because if there is vitamin B12 deficiency, then folic acid alone does not improve neurological signs and symptoms that occur due to vitamin B12 deficiency. And dietary management is also important. Diet rich in folic acid, that is the green leafy vegetables and fruits, they are very important. What are the consequences of untreated folic acid deficiency? Because the folic acid deficiency results in the megaloblastic anemia, ineffective erythropoiesis, there is pancytopenia. So it leads to various symptoms like weakness, glossitis, angular stomatitis, oral ulcers, and various neuropsychiatric manifestations can also be seen in the folic acid deficiency, like depression, irritability, insomnia, cognitive decline, fatigue, and psychosis. And the main reason is because this low folate and high homocysteine level, it is responsible for affecting the cognitive function. Now if there is increased intake of folic acid or folic acid megadoses are taken by the person, then there can be increased risk of colon cancer as well as prostate cancer. Now let's see the therapeutic role of folic acid analogues like sulfonamide, methotrexate, pyrimethamine. These are the folic acid analogues. We know that folic acid synthesis requires pteridine plus PABA plus glutamate, and it also requires dihydrofolate synthesis. Folic acid synthesis does not occur in humans, it occurs in microorganisms. Further, the reduction of folic acid to tetrahydrofolate by dihydrofolate reductase, it occurs in both humans and microorganisms. Tetrahydrofolate is further required for various processes like amino acid synthesis, purine synthesis, thymidylate synthesis. Now the sulfonamide is a folic acid analog which is structural analog of PABA, and it competitively inhibits this dihydrofolate synthetase, and it leads to reduced formation of folic acid, and it acts as a antibacterial drug because it is responsible for destruction of microorganism, and it does not affect humans because folic acid synthesis does not occur in humans. Now this methotrexate, it is the competitive inhibitor of dihydrofolate reductase. It is used as anti-cancer drug, but it acts on both rapidly dividing cancer cells as well as normal cells, and it leads to various adverse effects, and that's why N5-formyl tetrahydrofolate, which is also called as folinic acid or citrovorum factor, it is given to eliminate toxic effects of methotrexate. Another folic acid analog, that is pyrimethamine, it is the anti-malarial drug. So the sulfonamide, methotrexate and pyrimethamine, these are the folic acid analogs. So in today's video we have seen the folic acid, that is vitamin B9, its sources, RDA, then biochemical functions, deficiency manifestations, laboratory diagnosis of the deficiency manifestation and management. We have also seen the therapeutic role of folic acid analogs. So I hope this video will be useful to you. Thank you for watching and happy learning.